When you picture evolution, what's the first thing that comes to mind? For most of us, it's that iconic tree of life, right? Everything branching out from one single root. But what if that's not the whole story? What if there's another way to look at all the evidence? Some scientists are suggesting a totally different model, and we're going to dive into it. So on one side, you've got the classic tree of life. This says that every single living thing, from the tiniest bacteria to you and me, all traces back to one universal common ancestor. But on the other side, there's this idea of a forest of life. Now, the idea here is that life didn't start from one single trunk, but from many separate, distinct starting points, almost like a forest full of individual trees or bushes. Each one could change and adapt, for sure, but only within its own basic blueprint. So the big question is, which idea does the hard evidence really point to? And that's really the heart of the matter, isn't it? When you have these two massive competing ideas, how do you even begin to weigh the evidence? We need some kind of reliable filter, a way to sort the solid, high-confidence science from the stuff that's maybe more of an educated guess. Well, this is where biomedical engineer Dr. Rob Stadler comes in. He developed a framework for doing just that, and he got the idea from his work with medical devices, a field where the standards for evidence are, incredibly high, because people's lives are literally on the line. So here it is, his 6-point checklist for what he calls high-confidence science. Can you repeat the experiment? Can you measure the results directly? Was it planned out in advance? Does it do its best to avoid bias? And this next one is huge. Does it avoid unstated assumptions? Finally, are the claims it makes reasonable, or are they stretching the data? The more boxes you can check, the more you can trust the result. And this quote really drives the point home. In medicine, you would never accept low-confidence science, right? You expect and you require that the best possible evidence is backing up a procedure. So it kind of begs the question, shouldn't we apply that same level of rigor when we're talking about something as fundamental as our own origins? Okay, so let's take this filter for a spin. Let's apply it to some of the classic pieces of evidence that are often used to support that single tree of life model. And we'll start with a number that I bet you've heard before. Yep, that one. The famous 98.5% genetic similarity between humans and chimpanzees. I mean, it's often presented as the ultimate slam dunk evidence that we share a recent common ancestor. But let's run it through the filter. And here's the catch. Here's the context that often gets left out. That 98.5% number, it only comes from comparing the tiny sliver of our DNA that codes for proteins. We're talking less than 2% of the whole genome. So, what about the other 98%? According to synthetic chemist Dr. James Torr, this is where the whole thing just blows wide open. He points out that the other 98%, which people used to dismiss as junk DNA, is actually packed with critical information. And it's in that massive part of the genome, he argues, where you see these vast, profound differences between us and chimps. Now, this same filter can be applied to other classic evidence too. Take the fossil record. It's not repeatable, it's indirect, and let's be honest, the interpretations can be really subjective. And what about those genetic comparisons? Well, according to this framework, they often fail that unstated assumptions test. Because to make sense of the data, you first have to assume common ancestry is true. That's a built-in assumption that kind of short-circuits the investigation. Okay, so if the historical evidence is considered lower confidence, what happens when we look at science we can actually repeat and measure directly? What does high-confidence science from the lab tell us about what evolution can really do? Let's take a look. This brings us to the incredible Lenski experiment. Scientists have been watching E. coli bacteria evolve in a lab, in real time, for over 30 years now. That's more than 80,000 generations. To put that in perspective, that's like watching 1.6 million years of human evolution play out right in front of your eyes. And then, around generation 33,000, something really cool happened. The bacteria suddenly developed a new trick. They could eat a chemical called citrate. This was a big deal because their ancestors couldn't do it, and it was hailed as this beautiful example of a major evolutionary innovation. But what was really going on under the hood, at the genetic level? Well, when scientists analyzed the DNA, they found that no new creative genetic information had appeared at all. What actually happened was an existing gene got accidentally copied, and another gene basically lost its off switch. It wasn't a new invention, it was more like hot wiring a car. They rerouted a pre-existing system by breaking apart. It was a clever adaptation for sure, but it wasn't the creation of anything fundamentally new.
And this whole theme of limits isn't just a one-off thing. In another high-confidence experiment, scientists deliberately broke a gene in two specific spots and then just let trillions of bacteria try to fix it. Even after thousands of generations, evolution couldn't manage to repair just two simple errors. Instead, the process just gave up and deleted the broken gene entirely.
So, with these very clear, repeatable results from the lab in our back pocket, let's circle all the way back to those two big models we started with, the tree and the forest, and see how they hold up. Okay, so the single tree of life model really requires evolution to be this incredibly powerful, creative force that can build brand new functions from scratch. But what we actually see in high confidence experiments is a much more limited process. Minor tweaks, breaking things, loss of function. On the other hand, the forest of life model, with its separate starting kinds, actually predicts that change would be limited within those boundaries. The sources also point out this model might better explain something called orphan genes. These are unique genes in a species that have no relatives in any other known life form. For the forest model, that's not a puzzle. It's exactly what you'd expect if each kind started with its own unique genetic toolkit. So, after running all this through our evidence filter, what's the big takeaway? What's the central challenge this all presents? It all really boils down to this question. If our very best, most rigorous, repeatable science, the stuff we can watch happen in a lab, shows that the evolutionary process is highly constrained and limited, then how confident should we be in lower confidence historical evidence that claims it's an unlimited creative force capable of building everything from a single cell? It's really a question about evidence and what we choose to build our beliefs on, and it's definitely something worth thinking about.